thanks to the committee and Dr. Martin. So I can have the chance to share what we have done and I'm doing in living skin equipment, the establishment and applications. Mainly, why we developed living skin equipment, because we have the clinical requirements that to treat a skin defect or some chronic wound, which is intractable and not easy to heal. And basically, we should have the technical support. The one thing is, of course, the cell processing center, and technically, we should separate the cells, the epidermal cells and fibroblasts, and then culture and expand the cells so we can have the cell banks. So we use the cells to develop the different kind of artificial skin, such as epidermal sheaths, artificial dermis, or the living skin equivalent. Okay, I will try to introduce what we have done in this field. Okay, basically, we can harvest the skin sample from the patient, and then we use this base to separate the epidermal compartment and the dermis. And we use a trypsin to separate the epidermal cells and the culture and expand the cells. For the dermis, okay, we can cut it into small pieces and then to seed the pieces of dermis in the culture dish, and then waiting for the outgrowth of this fibroblasts and then expand it. Okay, here, I'm sorry, this slide is in Japanese that shows you how we make the epidermal sheet. Okay, first we can harvest the skin sample from the patient, and then to culture the cells, and then make a lot of cells and then store it in the liquid nitrogen. And then we can use the cells again, the first, the second, third time to make the epidermal sheets, and then we can apply the epidermal sheet to the wound on the lab. Okay, this slide shows you the epidermal sheet we have prepared for the patient. Okay, the epidermal sheet can be of different size, smaller or bigger, as we required, or in different size of dishes. And the right side shows you the histology of the epidermis. We see the epidermal cells stratified in many layers. It's kind of similar like the normal human epidermis. Okay, and it is good. And the process of the preparation is simple, and can make a larger area of this skin, can do it again, can be used as, and also cost is not so high. But the problem is it's too fragile and not so resistant to friction. And it cannot stay on the wound for a longer time. And here's the two cases. The first thing is a chronic ulcer for many years. So we apply this epidermal sheath on the wound again for 8 times for just a few weeks. And then, okay, the wound healed. And the other case is for the burn wound. From the picture, we can see it's deep second-degree burns. So usually this kind of wound may last for over 2 weeks and may result in scar. But in this case, we apply the epidermal sheet on the wound for several times, and in 12 days it healed. Usually, from the picture or from this course of healing, there's no scar formation. why this epidermal sheet is so effective in treating wound. The first thing we think that it can act as a biological dressing. It can provide a very good microenvironment for the wound to heal. And the other thing is the cells in the epidermal sheaths can produce different kind of growth factors compared with the monolayer culture of the cells, and the epidermal sheaths can produce much higher level of growth factors. And this slide shows that in the wound, there are different cells such as the endothelial cells and fibroblasts. So the epidermal sheath can produce different kind of epidermal cells, different growth factors. The growth factors can act on different cells. For example, EGF family can act on the epidermal cells, and the VEGF on the endothelial cells, and TGF-beta or FGF can act on fibroblasts. And then, but the epidermal sheet is very fragile and usually cannot stay longer on the wound. It's too weak. So we try to develop this living skin equivalent. It's composed of two layers: the dermal compartment and epidermal and epidermal compartments. Usually we use the type 1 collagen as a dermal matrix. So we mix the fibroblast in the collagen and to make the dermal matrix, and then we seed the fibroblasts on the surface of the fibroblast populated dermal matrix, and then we culture it in the air liquid surface. In this way, we find the keratinocytes differentiated and stratified and differentiated to make it more similar as the normal skin. Okay, this the picture shows you. Okay, the left one is the living skin equivalent. We composed with the type 1 collagen as the dermal matrix, this one. And right one is the normal skin. Basically, the structure is quite similar, and we notice that the epidermis is very well differentiated. We know that for the human skin, the epidermis must be well differentiated. If not, it is kind of a disease situation. Okay, and this kind of LSE can be made into different sizes. It's dependent on the well. For example, this one is 6 well, trench well. It's the small size. The LSE is about 1 centimeter in diameter, and this one we can make a bigger one that's about 8 or 9 centimeters in diameter. Okay, we also try to use this living skin equivalent to treat some diseases. In this case, okay, we find this RDEB patient. They have skin bullous again, and result in ulcer and scarring. And we apply for this kind of LSE on the patient. Okay, just one time. This LSE graft tape, and 7 days later, the wound healed. And here's another case, also for RDEB patient. Okay, you can see that, okay, this LSE graft tape and survive. However, we also find that, the LSEs, the above LSE is better than epidermal sheet in treating the wound. It has a better take rate. But also we found some shortcomings is about the basement membrane development. In this kind of LSE, the basement membrane is interrupted. It's not well developed. It actually influences the function or the morphology of the LSE. So we try to make an LSE which can composed of a better developed basement membrane. Also, we want to make LSE with more tough texture. Even the type 1 collagen LSE is very fragile. It's kind of gel. Okay, we found this material. It's from human beings, human amnion. Usually, it's disposed after the after baby delivery. The amnion had been used in clinical work because of its has a function of anti-inflammation and can enhance the wound healing. Also, we found that the basement membrane in the amnion were quite similar to human being in structure or molecularly. Here shows you how we make such LSE with the human amnion to improve the basement membrane. First, is the same as we did before. That is, we composed the fibroblast populated collagen gel, and then put the amnion on this gel, but with the epithelial side up. And then we see the keratinocytes on the epithelial side of the amnion, and then we do the airlift culture. Here shows us the new amnion living skin equivalent. Here shows you, okay, in the lower part is the collagen gel, and then in the middle is the amnion, and then his epidermis. We found that, okay, the epidermis presented better morphology, and the basement membrane, the lines is much better. Also, the electroscope showed us the basement membrane structure is much better. Okay, and this slide, we detected or stained some molecules such as keratinocytes, and E-cardoherrin and desmoglein, which shows that in the amnion class, LSE, the epidermis is better developed. Yes, better developed. The structure is much better. While in the LSE without amnion, the epidermis is still activated. And this slide, we try to stain some basement membrane structure such as type 4 collagen, type 7 collagen, or laminin 5, or the components of the desmosome such as integrin alpha 6 and integrin beta 4. We found that in the LSE with amnion, the basement membrane is better developed. However, since we have done this, we are not satisfied with this. So we try to make the amnion LSE without collagen. So at this side, in this slide, I showed you that in the matrix side of the amnion, we see the fibroblast, and on the epithelial side, we see the keratinocytes. So in this process, we saved some time, and we do not use type 1 collagen, and we develop the skin equivalent free of animal components like this. And the morphology is anyways good, and the basement membrane or the desmosome, the desmosome is well developed. And this slide, we stain the basement membrane with laminin 5, type 4 collagen, type 7 collagen. We see that it's very clear and linear basement membrane in this living skin equivalent. And then we try to transplant it. To transplant this new living skin equivalent in nude mouse. Here shows you the case. Okay, we made the skin defect on the back of the nude mouse and then transplanted. For 2 weeks later, okay, the wound healed. The wound healed and the living skin survived. And this picture shows you, okay, this is the amnion living skin equivalent. Here shows you, okay, here's the amnion still here. And we also try to use it in clinical work. Here, okay, we prepare the living skin equivalent with the amnion as the main matrix. Okay, the size can be different size. It depends on the dish. Okay, we can cut this kind of LSE different size and the shape like this, according to the clinical requirements. In this case, this patient has a giant nevus on the back, and the doctors removed the giant nevus and prepared the wound with the fresh granulation tissue and grafted the LSEs with the stem grafting. This is about 1 week after the grafting. We noticed that the LSE survived, and 1 week later, this LSE stems expanded and fused and covered the whole wound. And in the other case, it shows you the hand with the scar contracture. So the doctors separated the contracture and exposed such a wound, and then they grafted the wound with the amnion living skin equivalent. This is about 10 days later after the operation. We see, okay, the wound healed like this, and then the doctor makes the second and the third operation to separate, to say, fuse the fingers and grafted with this LSE. The above work, yes, we have done is mainly for the clinical application. Also, we know that if we want to do some skin experiment, we cannot always use the human or use the animal as a model. This is not convenient all the time. So this LSE, because it is composed of 2 layers of with dermis and epidermis, quite similar to the normal human skin, so we can use it in some skin research work. In the first one, yes, we try to transfer some genes such as GFP plasmid into the keratinocytes. Here, and we find, yes, the keratinocyte express the GFP genes. And in the skin wound model, this is the LSE. We cut it through the epidermis into the dermis. Then we investigated how the cells interact with each other in the healing process. Also, in this model, we applied some growth factors such as the basic fibroblast during the building of the living skin equivalent. So we found that these growth factors affected the differentiation or the homeostasis of the epidermis or the dermal cells. And all in all, yes, we have done such work in the living skin equivalent. However, if we want to do this work in a large scale or let more doctors or researchers use this model, we should do something to lower the cost to enhance the efficacy. The first thing we know that when we're developing the living skin equivalent, we need a different kind of culture medium. And we need the culture medium without the animal components. It cannot be too expensive, and they should produce the cells faster. So we try to develop some new culture medium to support our work, and also we want to develop the better and new dermal matrix to support this work. Okay, this is what we have done for the keratinocytes culture. The epidermis as a control group, it is commercially available. However, it is expensive. We developed a new medium, also free of animal components. We see that in the primary culture or the passage for culture, we find in the culture, it's a new culture medium, the keratinocytes presented a better morphology and with a higher efficacy. And for the mesenchymal cells, for example, the fibroblast, usually we use the amnion medium with the supplement of the 10% FCS. So in the new culture medium, we have a much lower concentration of FCS. However, the cells proliferate much faster in the conventional culture medium, and you see that the cell morphology also much better than the conventional one. And also this new medium is not only for the fibroblasts, it can be used for the other mesenchymal cells. For example, this is adipose tissue derived stem cells. You see the cells presented a much better morphology and proliferated much faster, and also it preserved the stemness of these mesenchymal stem cells. Also for the dermal matrix, we did some work. For example, we seeded the mesenchymal cells such as fibroblasts or adipose tissue-derived stem cells, and we stimulate the cells. The cells can produce and deposit the extracellular matrix like this, okay? And here's the histology, shows you the structure, and here the staining of the collagen or hyaluronic acid, and here shows us the electroscope shows us the outer structure. We can see the bundles of collagen, and we see the cells attached adhered to the bundles. And here, and with this fully cell assembled extracellular matrix, we mixed it to such a mixture of the ECM, and then we seeded subcutaneously on the back of the mouse. It's 2 months and 6 months. We can see that this matrix stayed and survived on the back of the mouse. And also, we found that in this self-assembled ECM, the cells functions much better because it also produces a higher growth factors such as HGF, VEGF, KGF, and TGF beta. And this higher level growth factors may also contribute. Okay, thank you so much.